Good evening, all. It gives me great pleasure to welcome all to the fourth session of the lecture series for propagation of statistics, education, and research as part of the outreach program organized by the Calcutta Statistical Association. We are delighted to have participants joining us from various institutions and backgrounds, united by a shared interest in the field of statistics and its growing relevance across disciplines. The Outreach Program is an important initiative of the Calcutta Statistical Association aimed at connecting with a wider academic and professional audience. We are especially honored to have with us today the President of the Calcutta Statistical Association, former Professor of Statistics, University of Calcutta, and former Pro-Vice Chancellor Academic at University of Calcutta, Professor Ashish Kumar Chattopadhyay, our revered sir. Before we introduce today's distinguished speakers, may I request the President, Professor Ashish Kumar Chattopadhyay, sir, to kindly say a few words about the Calcutta Statistical Association, its legacy, and ongoing activities, as well as the objective of the outreach program. Over to you, sir.
Thank you, Rana. So, good evening to the participants in India, and good morning to the participants in US and Canada. So, I am really delighted to be here in this lecture series, in the second lecture of the fourth session, to be delivered by Dr. Nilotpal Sanyal from Texas CI Paso. So you know that Calcutta Statistical Association in its activities, there are several activities. Some of them is very common and well known to all of you. That is the Triennial Symposium and the Calcutta Statistical Association Bulletin, which is regularly published. Now it is from SAGE, and many famous papers are published there. We have among us many say senior members like Nitista. They all know about these activities of the Calcutta Statistical Association. Now, for 2-3 years, we have started this outreach program, and I am really thankful to our young members who took the initiative to continue this series of lectures under this outreach program. In order to provide some new ideas which are coming in the area of statistics from the different institutes and universities all over the world, to make our members aware of this research activities. As you know, that presently we are in a path where the statistics community is really confused how to go ahead. What is the future of statistics under the pressure of data science and many other jargons, which are most of them which are mainly the part of the statistics. So I think this is the time when statisticians should start their activities in such a way to prove that without statistics nothing can happen. And I think our research activities also reflect such ideas and propagation. So I'm really, again, I thank you all for arranging such a nice lecture series. So Rana, you may continue. Thank you. You are muted, Rana, you are muted.
Thank you, sir, for those insightful words. Now, let me introduce the speaker of today's session, Dr. Nilotpal Sanyal. Dr. Nilotpal Sanyal is an assistant professor in the Department of Mathematical Science at the University of Texas at El Paso, where he also serves as the assistant director of the Data Analytics Lab. He earned his PhD statistics from the University of Missouri, Columbia, and has conducted postdoctoral research at Stanford University, UC San Diego, and Texas A&M University. Dr. Sanyal's primary research areas include Bayesian statistics, survival analysis, high-dimensional models, statistical genetics, and computational neuroscience. His research has been published in respected journals such as Nature Genetics, Bioinformatics, Neuroimage, American Journal of Epidemiology, Shankha, Statistical Methodology, etc. He is also the developer of multiple R packages. Dr. Sanyal is deeply committed to education and mentoring. At University of Texas, El Paso, he guides PhD and master students on statistics and data science research and has taught courses on advanced regression analysis, statistical computing, advanced data visualization, and survival analysis since his joining. In the past, he has taught at the International Statistical Education Center under the Indian Statistical Institute, Kolkata, delivering courses on data analysis, statistical methods, and descriptive statistics. He has also conducted various specialized workshops on general statistical methods and software, time series analysis, survival analysis, and machine learning methods while at Indian Statistical Institute and Stanford University. Over to you, Dr. Sanyal.
Hello, hope I'm audible to everyone.
Yes, you are audible.
So first of all, I would like to thank Sanjeev for inviting me for this speech, which I very happily accepted. And now I thank Shubhrata for introducing me. And I see my professors there, and I just send my respect to each of them. Let me share my screen. So do you know that there are approximately 100 billion stars in the Milky Way galaxy, even by conservative estimate? And do you know approximately how many galaxies are estimated to be in the observable universe? Can anybody say anything in a comment? How many galaxies approximately in that observable universe by current conservative estimate? Okay, so let me just go ahead and give you the estimate. So this is also 100 billion. So 100 billion stars in the Milky Way galaxy alone. And then there are 100 billion galaxies in the observable universe. You can think of the scale of the terms. But that's if that is not startling enough, it is interesting to know that our body, the human body, it contains more cells than the number of stars in the Milky Way galaxy. So there are in the average human adult body about 30 trillion cells, 30 trillion cells. And it moves around that figure. And not only that, all of those cells contains the human genome, which is the hereditary information that goes from generation to generation. Each of those 30 trillion cells pretty much contains that genome. Specifically those cells which contains a nucleus. But that's not all. Our body also contains many more organisms than we think. So if we really think about them, we can kind of suffer from an identity crisis, because our gut, what we call when we call a gut feeling, so our gut contains about trillions of microbes, what we call bacteria, microbiomes, and each of those bacteria have their own genome, their own genetic variants, genes, and etc. So our human genome only contains 21,000 genes. I'm going to show you soon later. But those microbiomes, they themselves contain more than 2 million genes. But in this talk, I'm just going to talk about human side. But it's interesting to see that within us, there are those microbes which are also part of our system, and they contribute a lot. So if we look at these terms, then it's mind-boggling that so many lives inside us. And then not only that, any two persons shares about 10 to 12% of those microbes, which means most of the microbes are not shared, and which means the microbe signature in our gut is just like our signature, like our fingerprint. It's very unique from person. It's very unique for that person and varies from person to person. So all of these things are very mind-boggling. But on the other hand, now let's just come to our human body. Now our body contains about, as I told, 30 trillion cells. But did you know that for example, our stomach linings, the stomach cells, they're only about 5 to 6 days old. The stomach linings that we had like a week ago is very different from what we have and maybe completely renewed. The cells in our tongues, our taste buds, they renew every 10 days. And then there are also other things. Our skin kind of replenishes itself in a month. Our lungs are like 5 weeks old. Our livers are a little more stable. They're like 3 years old, which means all the cells are replaced within like 3 years. Our bone structure is like 10 years old. Our whole skeleton, including the muscles adjacent to the bones, they kind of replenish in 15 years. Now it can be shown that every day a person kind of replenishes 300 billion cells out of the 30 trillion cells. And within 80 to 100 days, we have kind of like replenished almost 30 trillion cells, which is equivalent to the whole human body. So we are a very dynamic organism. We are not a fixed set of cells. The cells are continuously changing. But what keeps us stable is the forms. And how those forms are formed, that instruction is within our genome, within our genes. So today I'm going to talk about at that level. The title of the talk is Navigating the Biological Multiplex with the Torch of Statistics. So first I'm going to tell you about what is the biological multiplex that I mean here, which will include our genetic data, and from that genetic data, it will talk about the RNA data, protein data, and metabolome data. All of these are part of our biological system. And then I will say how statistics can help us learn about this sea of complexity, this labyrinthine gold mine that the omics data is. So with this much of introduction, let me welcome you and dive into this talk, and let's look at it together. So I will assume no prior knowledge on the omics and the statistical analysis of omics data. And I was told that this talk should be geared towards students. So that's how I designed it. So by the end of this talk, I will assume that most of you who don't have an understanding of omics or who don't have enough background on omics will have a solid understanding of what are the questions omics analysis tries to answer using different statistical and data science tools. So I will start with a very simple introduction, a layered introduction, and then after the introduction, I will tell a few statistical methods that are used to address some of the problems that arise in omics. Now, of course, in this short talk, it's not possible to go into the details, so I will refrain from using mathematical equations and models, etc. But I will just mention their names, what are some of the models used. And there are also zillions of models for different layered analysis, but I will just mention a few of them. And so that you get some idea about what kind of questions are answered. So starting with the cell, as I have already told, we have like 30 trillion cells, and each cell approximately is of the diameter 10 to 30 micrometers, which is like 100 to 300 of a millimeter. A millimeter is like the difference between these two fingers, and then 100 to 300 of this distance is the diameter of an average cell. Now, most of the cells contains nucleus at its center. Some cells do not contain. For example, red blood cells and platelets, they do not contain a nucleus. But for all those cells that contain a nucleus contains chromosomes. These chromosomes are like thread-like structures in the nucleus, which we can see in this figure. So this is a nucleus of a cell, and these are thread-like structures. Now, there are two types of cells in the body. One is called somatic cells or body cells, which makes most of the body. And the other is called the gametes, which is the sex cells, sperm and egg. Sperm in the male, egg in the female. Now, all the somatic cells, the body cells, contains 23 pairs of these chromosomes. We can see 23 pairs here, total 46. These 23 pairs are named chromosome 1 to chromosome 22, and then one pair of sex chromosome called XX in the females or XY in the males. Now, all of these 46 chromosomes are within this nucleus. And you can see that, for example, the chromosome 1 is the longest chromosome, chromosome 2 is the second longest, and it's ordered accordingly. Each chromosome, each of these 22 pairs appear in, they have like two copies, which we can see, and sex chromosome has either XX or XY. On the other hand, the sex cells, which are the gametes, they contain 23 chromosomes, because these gametes, sperm and egg, they combine to become a zygote, and then the zygote actually becomes a fetus, which develops into a baby. So the sex cells, each of them contains 23 chromosomes, half the number. Now let's look at inside the chromosomes what's happening. Now we can see that each chromosome here is like a structure here. Now this is called a double helix structure. And each chromosome is made up of a DNA molecule. It's one molecule, very long molecule, which kind of tied around proteins, which are called histones. These histones are like packaging around which this DNA molecule is coiled. And this structure is called double helix. It's like a ladder kind of twisted. Now, this DNA molecule is actually in the coiled form, but if it's stretched out fully, it will become almost like 0.7 to 3.3 inches, which we can really see. So that is pretty long. And such a long molecule is inside the cell. And just because there are these proteins called histones, that's why these molecules don't get entangled too much. Because if this gets entangled too much, our genetic information will be too much messed up, and we don't know what's going to happen. So these proteins, which are the packaging, they stop this long molecule from getting intertwined with other molecules and etc. Now, what's inside this DNA, this ladder? This ladder is actually made up of molecules called nucleotides. So this is like a nucleotide, A, etc. So this whole thing is a nucleotide. Now, each nucleotide has three parts: a sugar molecule, a phosphate molecule, and a nitrogenous base. And these bases are called ATGC: adenine, thymine, guanine, and cytosine. And these bases are what contains the genetic information that is passed from generation to generation. So our nucleotide is kind of synonymous with a base functionally. So sometimes they are called like base pairs. Now the human genome has like 3 billion pairs of bases. These are measured as pairs because these bases are in the two sides of the rung. And if we know one side, we know the other side. So if this side is A, then this side has got to be T. If this side is G, then this side will be C. So knowing only one side is enough. So this is measured as base pairs, and our genome has 3 billion base pairs. Now let's come to the whole picture. So within one cell, we have 46 chromosomes about within each somatic cell. And each chromosome is like a DNA molecule, and each DNA molecule is composed of so many nucleotides. And if we collect all of this as together, so here in this,
Nilotpal,
yes,
sorry for the interruption. Could you
yeah.
please make your presentation full screen?
Oh, okay, all right.
Yeah, now it is perfect.
So we have all of these chromosomes inside our body, and this whole collection of all of these chromosomes with all of their DNAs, this is called a genome of a person. So it's a human genome. So in a human genome, we have like 23 pairs of chromosomes. Now this genome is within each cell. So we have like trillions of cells. Within each cell, there's this genome, so hereditary information. If few cells are destroyed, the whole information is not destroyed. It lives onto the other cells. And each genome contains the entire set of hereditary instructions for building, running, maintaining, and whole organism. Now, what is a gene? A gene is a part of this genome, a functional part of this genome, which is enough to code for a functional product. And the functional product is either a protein or functional RNA. Now, proteins are the workhorse of our cell. They run all the works in our body, actually. So the DNA is actually the blueprint of the plans, which makes our body in a particular way, and an elephant's body in a particular way, and a monkey's body in a particular way. For example, we all eat banana, and then a monkey also eats banana, and the elephant also eats banana. But that banana becomes an elephant in the elephant body and becomes a human part in the human body. It's because the plan on how the parts will develop, that's encoded within the DNA, that's the blueprint. But the actual work, so according to the plan, that is carried out by the proteins. So they are the workhorse of the whole body. And a gene is a segment of the DNA that is enough to code for a protein or something called a functional RNA, combinedly a functional product. And that's why genes are called fundamental units of heredity. Now, human genes are commonly approximately 27 base pairs long, but they can be up to 2 million base pairs. This is one example of a gene. This is like TP53 gene, which is often voted to be the most popular gene. Here I included a link for some of the most popular genes. Now, this is just a part of genome consisting of many nucleotides. They together code for a protein or a functional RNA. If we divide the human genome into different parts, it will have about 1 to 2% only these protein coding genes. And there are 5 to 10% of non-coding genes, which don't code for a protein, but codes for a functional RNA, which is like RNA. And then there is the non-coding DNA, which compose most of the genome. 90 to 94% of the genome are not coding for proteins or RNAs, but they have their different functions like regulatory roles, structural roles, etc. So they include promoters, enhancers, silencers, all of this. All of these are important, but only 1 to 2% make proteins. Why proteins are important, as I already told. Now we'll see that what is the process of gene expression. Gene expression is a name of a gene converting into a protein. It happens in 2 different stages. From the gene, from the DNA level, it first becomes an mRNA, messenger RNA, through a process called transcription, and then it becomes a protein through translation. So this transcription and translation make the whole gene expression procedure. Now, these terms are extremely important in understanding omics data because for each of this layer, there are statistical measures and there are questions that you answer with statistics and data science. So the transcription happens through something called RNA polymerase, and it's controlled by the regulatory elements which are in these parts of the genome. And the translation happens through ribosomes, and these amino acids are assembled together into a protein. 3 consecutive nucleotides, like maybe ATG or GTC, etc. 3 consecutive nucleotides code an amino acid. And there are 20 amino acids that are possible out of that. And out of those amino acids, different proteins are made. So the next important concept is mutation. A mutation is like a permanent change in the DNA. It's like adding a random noise to something. When we generate data, we generate something systematically and then add a random noise. So that is like the mutation. It's a random change in the DNA sequence, which has become kind of permanent over the time. Now, mutation can be classified into different ways based on the origin, based on the DNA sequence, based on something called a copy number, or structural changes, and based on protein effects. I'm going briefly over some of them. So the mutations that are present in the germline. Germline means when the sperm and oval cells meet, that becomes a zygote. That is the germline cells. So any mutation, any random change present on those germline cells, they actually comes into the baby, because baby starts with one cell, and that one cell multiplies into millions and trillions of cells over the lifetime. So if any mutation is contained in that one cell, it becomes part of every cell in the body. So those mutations that comes from the parents through germline is called germline mutation. And there are somatic mutations which a person acquires during his or her lifetime that are not part of germline, that is somatic. You can also classify mutation according to change of one nucleotide only. So if you can see here, A became G or C became T. So change in one position by random chance, by random variation, then it's called single nucleotide variants, SNV. If AGT becomes AGTT, so one T is inserted, it's called insertion mutation. And then AGT becomes AT, G is deleted, it's called deleted mutation. And then larger chunks of nucleotides like XYZ, their copy number changes. So it was like XYZ became XYYZ. So one whole set of nucleotides Y is kind of inserted in between. These are called copy number changes. And there are different types. There's inversion. One kind of sequence is completely inversed. That is called inversion mutation. One's translocation, which means one part of the genome moved to another chromosome altogether. That's called translocation. And then based on the protein effect, if the change has no effect on the outcoming protein, it's called silent mutation. If it has change in the sense that the protein is changed to some other protein, then it's called missense mutation. And if no protein is produced at all, the protein generation actually stops because of the mutation. That's called nonsense mutation. So these are different terms of mutation, and you have statistical procedures to detect these mutations. Here are some of those what I told in terms of figure. So here, this is the original sequence, which is in the reference genome. A reference genome means a population genome or a genome which is established based on many persons or based on ancestry. Now, if in a particular person, a sample genome, this part is not there, then this is a mutation, this part is gone. This is a deletion mutation. Similarly, this is an insertion mutation. This is a duplication mutation. This part got duplicated twice. This is a inversion mutation. This part got completely reversed, etc. And then these are examples of missense and nonsense mutation. So the corresponding protein here, it was supposed to be this, but it became this. That's a missense mutation. And this actually stops the making protein altogether at this stage because of one change here. G became T. So this is a nonsense mutation. Now, all of these things are compared through like genetic markers. So genetic markers are positions in the genome that are compared across subjects or individuals to identify that those positions, to align their genomes. And the most important marker is single nucleotide polymorphism, or SNPs, which are like a variant which is more than 1% prevalent in the whole population. One SNV, which is more than 1% prevalent. So this is one mutation which is kind of present, which is different from the reference, but which is present in more than 1%, which shows it is kind of common enough. But there are also others which are less than 1%. They're not considered common. And most of the rare ones is actually suspected to be the causes of diseases, genetic diseases. So there are also other markers which are called tandem repeats of microsatellites. They are multiple nucleotide long or multiple base pairs long. Next, the concept of allele. An allele is a variant of a gene. So here we saw the variant of one nucleotide. A variant of a gene is a whole sequence of nucleotides which codes for a protein. Now, if something is changed within this sequence, then that changed version is called allele. Now, we already have seen in this figure that we have like each chromosome comes in two pairs. And now, so each chromosome contains the same genes. It's supposed to contain the same genes. Now, if the same gene here is one version, here is another version, and these two versions is called alleles. They're supposed to be same, but they're not same, then they are called alleles. If they're same, then they are called, then this position of this gene is called homozygous, come from the word zygote, homozygous. But if they're not same, they're called heterozygous, and each version is called an allele. I know that each part comes from one parent. So this part comes from the father and this part comes from the mother. So this, now if they're the same, then they're homozygous, otherwise they're alleles. So these are all genetic changes that can happen and name of different genetic variants. Now a few more words about alleles. An allele can be dominant, recessive, or codominant. Dominant means if this multiple variations, now you consider two variations. If the one variation is such that if it's present in only one of the chromosomes, it will bring out some kind of outside property, maybe black eye or blue eye, whatever. That then it's called dominant. Now, if an allele is such that it needs to be present in two copies to bring out a particular change, then it's called recessive. It's effective, but its power is a little less. So it needs to be in two copies. And if the alleles are such that if one copy of each allele is present, then kind of an intermediate quality is produced, then that's called codominant allele. So these are all different types of alleles. And the collection of all alleles of a gene is called a genotype. So for example, if a gene has two alleles, small a and capital A, then a genotype is simply a collection of these alleles, AA, which is written together. Now, a few more words about other genetic levels. So epigenetic changes. Epigenetic changes are changes that does not change the actual DNA sequence. So in this figure, you see this actual DNA sequence. These are not altered, but something other comes out from outside and they tags into this DNA sequence, like these molecules. This is called a methyl group. This methyl groups comes from outside and tags along with this, changing the function of some of the genomic forms. So these changes are called epigenetic changes. Another type of change is called histone modification. So we already seen that histones are the packaging where this DNA is packaged. If some part of the histone is damaged, then some part of the DNA may be not accessible. So we cannot use the information there. So these are all epigenetic changes that modifies the effect of the genes and genomes. Now, here are a few of the epigenetic changes. Next so we have seen like the genes, and then genes are part of the genome, and then the genes through transcription, they become proteins or functional RNAs. There are different functional RNAs. And then we have seen that how those changes can be modified by these epigenetic changes, which is like DNA methylation, a methyl group coming and joining, or histone modification, which is a damage in the histone packaging. Now, other than that, in this whole procedure from the genome to the RNA level to the protein, this whole chain of reaction, this reaction is also moderated by many other things, and this reaction also produces many other byproducts. All of this together are called metabolites. So this is a picture of a human metabolite that contains various different types, various modalities of molecules. But the common feature is that they have to have very small molecules. typically having molecular weight less than 1500 Dalton. Remember, DNA is a very large molecule, but these are very small molecules, but they take part in the procedure, or they are the byproducts of the procedure. And this includes, for example, primary metabolites are like glucose, ATP, amino acids, secondary metabolites, plant alkaloids, antibiotics, metabolites that are produced inside the body that are like hormones, neurotransmitters. We all know these names, but these are all metabolites. And metabolites that comes from outside, like drugs, toxins, different food-derived molecules that comes from outside, or the pills we take, the antibiotics we take, all of this. These are all the metabolites. So now you will get an idea of what an omics is with this background. So omics is a collection of all of these fields. So it starts with the basic level, which is the genomics, which is the study of the genome. Genome is a complete set of DNA in the chromosomes inside a nucleus, as we have seen. And each cell contains the whole genome. Then there's epigenomics, which are the complete set of epigenomic modifications, which is epigenome. Analysis of epigenome is called epigenomics. Then we have seen that the genes become transcribed into RNAs. So the complete set of RNA inside a cell or tissue or organism is called the transcriptome, and its analysis is transcriptomics. Then the RNA is transformed into protein. The complete set of proteins within an organism is called proteome, and it studies proteomics. And then all the molecules that assist in the process or that are byproducts of this process, they form the metabolome, which is very diverse and very dynamic, changing all the time. So if I'm taking a breath right now, my metabolome is changing. So something is changing there. So this metabolome, the study of that is metabolomics. So from genomics to metabolomics, these are the different layers of biological data. And the study of all of them are part of the omics. And finally, the visible characteristics of our body, for example, hair color, eye color, whether we have a disease, what we can see from outside or measure from outside, these are called phenomic. They're called like phenotypes. And the collection of all of them is called a phenome. And their study is phenomics. So together from genomics to phenomics, this is a whole gamut of omic studies. And this is a very complex study because it contains all the functions that our body performs and all the things that we do. The answers are mostly inside it. But then this omics is also modified by our behavior, our lifestyle, etc. But this is a whole picture. Now, once I have established the picture, now you can, of course, understand from this what can be some of the challenges in omics and analysis, thinking from statistical point of view. For information, a full multi-omic study, so one person's genome, transcriptome, proteome, metabolome, if we collect all the data together, they would be like 1 TB of raw data, 1 terabyte. So for one person, we generate 1 TB of data. And it's without the phenome, it's just the genome to metabolome. If we add the phenotypes, it will be even more. So for many persons, we can see the size of the data. So one problem is, of course, high dimensionality. There are thousands, millions of variables, millions of SNPs, thousands of genes. So we have like 21,000 human genes. And then thousands of proteins, thousands of metabolites. It's important that although this 21,000 genes go on to make proteins, because of something called alternative splicing, the number of different versions of proteins is actually like 2 million. Because one gene can become protein in different ways. I'm going to come to that later. And there's also a lot of noise and variability in the data. The noise because all of these things are measured through different scientific machines, and they have the technical noise. And there's also natural biological variation from person to person. Missing data often occur in the proteomic and metabolomic data because of the way they are collected. And then multiple testing, of course, comes because when we are testing something, it's the association of some kind of disease with maybe one gene, and then there are like 21,000 genes. We're testing the association of some kind of disease with each of these genes. So you have to take into account multiple testing conditions. So this false discovery rate based adjustments has to be made, or bond-friendly adjustments, all of these multiple testing adjustments. Finally, integrating data from different levels, genomics, transcriptomics, proteomics, metabolomics, these are all different levels. How to integrate them? That's a challenging problem. so that they're meaningful. Data normalization, because all of these things varies from person to person, and because of the way the data are collected, because of instrumental error, or because of different batches of data collection, you have to normalize these different data from different batches or different persons so that they're comparable. So this is an important problem. Interpretability, once again, how to interpret different omics data, how to connect them. Scalability, of course, whatever method you we devise for analyzing such data, are they scalable or not? It's very important. And finally, there are very complex interactions between genes. So gene-gene interaction or protein-protein interaction, they may not be simple linear, they may be non-linear, very complex interactions. So how to take into account them? So these are the challenges of omics data analysis, and which you would need to address if you do research in this field. Now with this, I will now try to summarize some of the approaches of different layers of omics analysis. I will pursue the problem through posing questions, because each question can be answered by a zillion number of methods. There are a zillion methods in the literature. I don't have scope to go over each of them. So I will mention a few of those methods, the basic methods that are established, but then I will just see the questions. I will put in front of you the questions so that you can think in terms of the questions. So let's first think at the genome level. So we have different, inside our cell's nucleus, we have different chromosomes, and the whole collection of all chromosomes contains our whole genome. How to identify genetic variants like the SNPs, or the insertion or deletions, indels, or the copy number variations, CNVs, in an individual genome by comparing it to a reference genome. So when a machine collects a person's genome, they take a bunch of cells and then they measure it through some method. What they do is they compare that person's genome with a reference genome, which is already established. Maybe a reference genome for all human beings, or a reference genome for a particular race, or some ancestral background. So they compare that one individual's genome with the reference genome, and the individual genome is read as different, this short reads, as you can see here. How to align these reads with the reference genome to identify the positions, that is called the problem of variant calling. So these are different variants, SNPs, indels, CNVs. And how to call the variants means how to exactly identify them, comparing the reads with the reference genome. There are different methods for that, the Bayesian or likelihood-based scoring methods, which assigns scores to each of these reads. And the scores kind of determine how close they are to the reference genome corresponding position. And based on that scores, they're called. There are pile-up and depth analysis methods, where they stack all the reads that part of the same genomic position. And then based on that stacked number, they kind of etch out an evidence which is modeled using some methods. And so this is a pile and depth analysis. There's different methods for CNV detection. CNV is a large portion of the genome which is kind of messed up, and they have their own different methods based on like hidden Markov models or different segmentation algorithms. For example, in the hidden Markov models, they assume that the actual CNVs in the reference genome, they are like hidden states, and then how much from which state actually the individual genome CNVs come. So that's determined based on hidden Markov models. These are applications of statistics for variant calling. Next, what other questions we might have based on the genome? Next is how much I'm doing in time? 8:48. Okay. So we have different other questions like which genetic variants, for example, this is a variant, a SNP. Which variant is associated with a particular trait, maybe our blood group being A within a family? Or what are the different possibilities of like dominant, recessive, or codominant patterns, like blood group can be A, B, or AB. So if your father's, it is known that if your father's blood group is A, mother's is B, then your blood group is going to be AB, because this is like a Mendelian trait, and this goes on by the family. So this type of associations, or which genes, so these are two genes, for example, which genes are co-inherited with the disease. So when we have a disease or trait like an A group, A blood group, which genes are co-inherited with that same group. So these two are inherited within this case, within that family. So this type of family-based analysis, it's called genetic linkage analysis. So it links persons within a family. Now, these methods were more famous before the sequencing methods came into the vogue, but these are for the historical background I'm telling. And there are different methods for analyzing this data, which means answering these type of questions. How do you make these associations? So you can use parametric methods like logarithm of odds to assess linkage under an inheritance model. So these are three different inheritance models: dominant, recessive, and codominant. Already I told. And so you model like the odds of under linkage versus no linkage. Or you do variance component analysis modeling, proportion of trait explained by each genetic sharing. These are parametric analysis. If you don't assume a model like a genetic recessive or any kind of model, if we just go for a non-parametric analysis, you can simply compare whether siblings with the same trait shares the same alleles. So depending on the number of shared alleles, you can develop non-parametric methods that can test for this linkage, genetic linkage. So this is one set of problems. Next comes another set of problems, which is instead of being within a family, it's within a whole population. So these are like population. Similar things you can test in a population, like which SNPs or which variants are associated with a particular trait. For example, the hair color. Now here, hair color is considered a complex trait. Complex trait means it is not the outcome of change in one gene, but multiple genes, maybe many genes affect these traits. So these are called complex traits. Compared to that, this blood group ABAB, it's like a simpler trait, and it's called a Mendelian trait. So Mendelian traits came earlier in the history, and they're simpler traits. The complex traits like eye color, hair color, our height, weight, etc., they're determined by multiple genes. So, and these complex traits are measured in something called genetic association analysis. So this came after the linkage analysis, and these are the type of questions they address. What are the SNPs that are associated with, for example, hair color in a population? Or what are the SNPs that are associated with hair color in multiple populations? Or what are the many SNPs that affect this hair color within one population? Or what are the environmental factors like sunshine or smoking or leading a good lifestyle? All of this affect the expression of this gene, and together they affect the hair color. So these are called genetic variables. These are environmental variables. What are their interactions within a population? All these are questions are answered in genetic association analysis. And the statistical tools used for that are different types of tests. For example, Fisher's exact test, chi-square test. We can always do a permutation test to test the association of different variants with different traits. So a variant is this, and trait is a hair color. And we also have to take into account the multiple testing corrections, as I mentioned before. Other methods, little more complex methods, linear regression. If you have covariates in your model, you can include it in a regression model, linear regression, logistic regression. If you have kind of clustered data, there are dependencies between a set of SNPs or a set of persons, you can consider mixed effect models. And then there are modern machine learning models, random forest, support vector machines, many other models that can handle non-linear relationships and interactions between those SNPs or variants. There are Bayesian models of some of these extended to the Bayesian part. Now here you have to, as there are many variants, you use a principal component analysis or similar methods to remove the confounding effects. So if there is a confounding effect in the population because of maybe older persons tend to have hair color more whitened, while the younger persons have not so whitened. So there is a basic difference between old and young persons. So take into account this difference, which acts as a confounding effect. You do kind of principal component analysis, which takes into account them, and you include a few principal components into your study to take their effects into account. So these are some ideas about association analysis. This genome-wide association study is an association study of the entire genome of a human genome. And the data on a single whole genome sequence from one person is about 100 to 150 GB in raw format. Okay, and I already mentioned some of the pattern exploration methods or dimension relation methods like the PCA. There are other methods like different types of clustering you can use. There is this partial least square based discriminant analysis or T-distributed stochastic neighbor embedding. These are all methods to kind of reduce the dimension to a kind of manageable dimension. Next, how to compare the methylation levels. So as I told, methyl groups are part that comes and tags along with the genomes. How to see what are the difference in methylation level between two persons. So it's simply a differential methylation analysis. The simple tools is like t-test, where you're kind of comparing two groups, or if you have multiple groups, you can compare them with ANOVA, or if you have covariates, you can use linear models. These are the basic methods, but there are, of course, a zillion of methods of this, but all of them are trying to answer this question, how methylation levels differ. Next question, how to identify gene structure elements. So in the DNA, there are these parts of the gene which are actually important, which convert into proteins. They're called exons. There are parts of the gene which are not. They are called introns. These introns, or they are promoter elements like this, or enhancer elements. These are part of non-coding genome. How they have their roles. They kind of modify the gene expression. How to identify the position of these non-coding elements. So where the transcription actually starts. Where is this promoter element? Where is this enhancer element? This is called structural genome annotation and regulatory element identification. So you identify the structural items. These makes a part of the genome. Where are they? And where are the regulatory elements? They also regulate the gene expression. So where are the positions? And once again, you can use hidden Markov models or different Bayesian network models to identify them. Next, now we come from the DNA level to the RNA level, the transcriptomics. How am I doing on time? 8:56. Sanjeev, can you tell me how much time do I have?
5 to 7 minutes.
Next is transcriptomics. Now, as you have some idea of genomics, now the transcriptomics ideas are pretty similar. There's some differences. Transcriptomics means transcriptome is a collection of all RNA, and how this RNA data is collected, that has changed over the years. Our earlier methods used like northern blotting or RT-PCR. After that, in the 1990s, we have this microarray-based methods. And then currently we have the next generation sequencing methods, which are called RNA sequencing methods. And the latest version is single cell RNA sequencing. So this version is actually sequences RNA, all the RNAs within each single cell of our body. So we can capture differences between each cell. And so you can understand that this RNA was based on a collection of cells, but now it's fine-tuned to each single cell. So we have a huge amount of data from scRNA sequencing data. Now, what are the questions? Which genes are active, which are not active? This is called gene expression profiling. You can measure this in terms of the RNAs in different ways. How can you make sure the RNA data is reliable? Because there are like millions of reads, and how to understand which are nonsense reads, which are not. There are different statistical methods of quality control and preprocessing. For example, normalization using different methods called TPM, transcripts per million, or TMM, transcripts mean, trim mean of n values, or different K methods per kilobase million. These are all methods of making the data comparable across subjects, across different batches. Then how do we perform pattern recognition, dimension reduction, as before using PCA, TSNE, or such methods. Next, how can I identify genes which are expressed differently? So these are different genes, these are different samples. Each samples mean different persons, let's say. But within each person, not all the genes are active at one time. How to understand which are active, which are not, or in other words, how to find the difference in the gene expression. You can use simple tests like t-test, permutation test, ANOVA linear models. You can also use count-based models like negative manual GLM, and then LRT world test to test for the difference of gene expression. And of course, there are Bayesian models. Finally, in the transcriptome level, there is a problem of alternative splicing, how one gene is spliced into different proteins. So this can be tackled with different statistical methods as well, likelihood and Bayesian methods. I don't have time to go over the methods, but just to give you the idea what do they do. So one gene can become this, another gene can become this. So this red portion is missing here. And then another gene can become kind of little reorganized. So the outcome is different version of the proteins. Actually different proteins. This is called alternative splicing, how to detect them. That's one important set of problems. And then how can each gene, the change in a gene, affects a biological procedure. A biological procedure is called a pathway. A biological pathway. Now, each gene kind of might change the pathway function or may not change. How to understand whether the changes in the gene expression is changing the pathway function? We have different types of analysis called overrepresentation analysis, gene set enrichment analysis, set-based methods, modeling-based methods, a network method, text mining methods. All of these are trying to say whether change in the gene expressions between two persons are actually affecting their pathway or not. So pathway is a biological procedure. For example, I'm breathing, that's part of a biological procedure, a set of reactions. Whether this set of reactions is being changed by one gene's change. That is pathway evaluation. And then finally, how genes correspond with each other. How do they interact with each other? This is called co-expression analysis. If two genes are expressed, if one gene is expressed in two different persons expressing in two different ways. So this is kind of a correlation map with which we can see these genes that are connected with ages, they're correlated, others are not so much. So these can be also analyzed using correlation methods, clustering methods, network-based methods, and the matrix factorization methods, which factorizes a huge genome matrix into smaller chunks, which are kind of independent Bayesian methods. These are all examples. The similar methods are for the proteomics level. We have also thousands of proteins, how to read them. This is called the mass spectrometry is used to read the proteins as this reads and how they're organized, and then how they're normalized to remove batch effects and how to make them comparable across samples, different normalization methods based on different procedure like total ion or quantile, etc. And then how can we compare the levels of protein in two persons, differential protein abundance, just like differential gene expression, we can use t-test, ANOVA linear models, and then advanced Bayesian methods and other modeling methods. How can we see post-translation modifications? The protein, after they're produced, they can also modify, and that's called post-translation modifications. And so there are like 1 million modifications that comes from 20,000 genes. How to identify those modifications and how to quantify them. There are different statistical methods for this stage. And how proteins interact with each other, protein-protein interaction network, that's a huge topic. And how to analyze this network to understand which are central proteins, which proteins are more connected with others, which are kind of not far-flung, lying at the far-flung corner, not much interactive. So finding communities within these protein networks, there are a bunch of methods for that based on different statistical models. And finally, which biological pathways are affected by protein changes, just like the gene, they're the similar set of methods. Similar methods for the metabolites, which are also thousands of molecules, similar to how they're detected and how they're normalized, how they're quality controlled, how compared between different persons, and then how they interact with each other, and how can you get predictions based on them, like whether this disease is connected with this change of metabolites or not. They're beginning from simple methods like t-test, ANOVA linear model, to advanced methods like random forest boosting, then deep neural network methods, different graphical models, all are used to analyze this genome, this omics data. This is another picture with showing pathway enrichment, pathway interactions. So, and finally, you can combine all of these omics from genomics to metabolomics. How to integrate all of this data together? That's an interesting, challenging problem. So for one person, you have genomics to metabolomics, all of this data. You can do individual layer analysis, but then how can you combine them together? This is called a systems biology approach, where you combine all of these things together harmonically to make meaningful conclusions. And these are a whole set of whole niche of analysis for which there are many methods. This is pretty hot topic these days. And they are like unsupervised factor analysis methods that finds out inherent factors in each level and then try to find out which are the common factors in different layers. There's supervised predictive models, variable selection models that are geared towards selecting a bunch of variables at different stages, and the network and graph-based models that kind of finds out communities, neighborhood, etc., and their connectedness. Clustering, by clustering models, they cluster the samples and the genes together, samples and the variants together by clustering models, and then deep learning hybrid models that take into account very complex and nonlinear interactions. Now, I will finish with just showing you a snapshot of how a particular data looks like. I don't have time to go over an analysis, a detailed analysis, but this is one particular data that I took from a website called TCGA, the Cancer Genome Atlas, that contains a lot of genome data. Not all of them are free, some of them are free. So this is sequenced using this Illumina platform, which is a software name where the sequence, like the genome, is read. Now the mutation data, this is how it looks like. So I read the file used in the R, I read it, and then the dimension is like this. So for 800 persons, we have mutation in about 5,000 genes, and they're like binaries. So this gene has a mutation or not, 0 and 1. This is a binary matrix. That is a binary data for mutation, whether mutation present or absent. And you can see that this can be a sparse matrix as well. So most of them can be zeros and some of them can be 1 only. Next, in the CNV's data, in the copy number data, it can be for like here for 1,000 persons, and for this much genes, we have the copy number, which is read in the log2 ratio scale, which you can see this is like a continuous variable. So copy number variation it's a continuous matrix of such large dimension. There's methylation data. This also measured at some kind of measure called beta values. Here it's standardized with kind of centered with subtracting 0.5. They are also continuous values for each person. These are person ID, and these are gene names. For each person, each gene, we have a methylation value. Similarly, we have at the RNA level, we have the RNA sequencing reads, which are like continuous data, or which can be also count data. Now, in the sequencing methods, they originally come as count data, but then when you normalize them, when you kind of adjust them for different things, they become continuous, and then you can fit continuous models. So they also, for different persons and different gene, we have different RNA sequencing expressions, RNA-seq data. And then miRNA data similarly. And finally, at the protein level, we have protein data, once again, for each person and each gene, because each gene corresponds to one protein, and because of alternative slicing, we get also millions of proteins. But here we focus on the gene names. So these for each gene, we have a protein expression, which is given by this, which is once again measuring how abundant in the protein is that protein in that particular person. It's a continuous measure. So these are a few examples. And finally, we have a clinical data. For all of these persons, we might have all of this omics data. And then we also have like the person's histological type, like what is this person's gender? What is this person's, for example, this data is a glioma data, which have a particular kind of disease. They have different types of glioma. What are the persons having those different types of glioma? Like these are different names, and the ethnicity of these persons. So these are like phenotypes, phenomes. So we have all the genotype variables and we have the phenome variables. Now, so you can see the complexity of this data. Now, how to analyze all of this data, how to find out patterns, how to find out interconnections. This is the whole analysis of the genome. So I think I will finish my talk here. In this short talk, it is impossible to go over into too many details, but I hope I have given a brief enough introduction, a brief enough overview on the whole topics, so that you can kind of understand what are the questions this omics analysis is trying to answer, and what are the basic statistical approaches that are used here. And then under each approach, there are zillions of methods that are coming out every few years. And so this talk was geared towards students. So I would assume that this is not the end, but rather end of a beginning for the students who are interested in genome analysis, who are interested in omics analysis and using statistical tools for that. they are welcome to explore more along these lines. Now, if you have any questions on any of these things which I cannot answer, which is not possible to answer in the short time, or if you want a copy of my slides, or if you need more resource on that, please send me an email personally, and I would be happy to send you feedback personally and send you the information. So thanks all of you for your time and attention.
Thank you, Dr. Sanyal, for your insightful presentation. Is there any question? So in the chat box, there is a question from Dr. Sanjeev Gupta. Any specific R package is available for exclusive for such type of study?
Yes, so there are many packages for omics analysis, which I did not include here because they will make the space longer. So there are packages, of course, there are many R packages for different types of analysis, but there are also individual standalone software for data conversion, for doing different types of preprocessing, and many other things. So yes, there are packages. Many of the packages are kind of in the bioconductor instead of the CRAN repository. They are in the bioconductor repository. Packages, there are also other standalone softwares that can be used for genome analysis. So if you're interested, I can, if you just send me a personal email, I can point you towards some of the softwares, because there are many, so it's difficult to tell in a short talk.
Is there any other question?
Sanjeev, I have a small query. Can I ask?
Yes, sir. Sure, Shishda.
Yeah, Nilotpal is a very nice talk. Congratulations for such a nice presentation. Actually, my point is, I think the students also will be interested in this point that you mentioned different statistical techniques, supervised, unsupervised. Now, for some of the techniques, their Gaussianity is important. Now, whether such a complex data set, whether Gaussianity is maintained or not, that is my question.
Yes, that is a very good question. So when you are using linear models, which are used in some of the established softwares, they of course assume Gaussianity, but that may not be the case always. But some of the established software which the actual biologists want to use, they sometimes look more at the interpretability. So they use simple software that assumes Gaussianity or very simple distributions, but that may not be the case in practice. So from a statistician's point of view, the distribution is very important. So of course, there are modern methods based on various non-parametric methods that are also used, and that you are also free to use. But for some of them, there are packages, of course, but there may not be established standalone softwares. But of course, if you cannot assume, if you don't see it's Gaussian, there is no point feeding a Gaussian model just for the sake of simple analysis. But there are non-parametric methods like different types of normalizations, which are based on the number of counts or the comparing the number of shared alleles between siblings. These are all non-parametrics. And then, of course, you have random forest boosting. All of these methods have been developed for each stage of omics analysis. And then you can fit neural networks with different types of activation functions. All of these have been explored and implemented in some paper. I don't have scope to go over all of them, but yes, there are many non-parametric methods as well which don't assume Gaussianism. But it's an important point, and the traditional ones generally assume Gaussianity, but you don't need to suppose that.
Thank you.
Is there any other question? Actually, like the data.
There's one question in the.
Does there anything related to circular association in when studying genomics data?
So what do you mean by that?
Circular association in studying genomics data.
Circular association studies. So do you mean whether there are redundant information that is being used in analysis? for of course the genes kind of interact with each other, and the proteins also interact with each other. So if you uh, by circular association, if you mean that if we are using the same information twice to predict something, is this what you mean?
You may unmute yourself and okay. Yes.
Oh, okay, yes. So yes, so remember that the genes go on to make RNAs go on to make proteins. Now, if you're analyzing the effect of proteins on some biological process, you can use the proteins, of course, but if you also use the genes along with the proteins, then that might be using redundant information. But you need to consider that from one gene through alternative splicing, different proteins can come. So it is one thing to associate a gene with a disease, and a little different thing to associate a protein with a disease, although the protein came from the gene, but the protein variations are many. So depending on which variations are more predominant, you can run a separate layer of analysis as well. So as I said, the interactions are very complex, and from a biological point of view, you need to understand what you're doing. It's not simply to get the data and then put into some statistical run some statistical function on that. You need to understand what preprocessing you need to do to make things comparable and meaningful, and then whether there are redundancies which you need to take into account in the beginning, and how to correlate data from different omics layers. I hope I gave you some satisfactory answer to that, if I understood you correctly.
Is there any other question? This is actually a very vast topic, and in this very short time, you have described in detail about the concept and the background, and you have briefly described all the methodological aspect to deal with this genomics data. So in this very brief time, the technique you have described is truly appreciated. So let us thank the speaker, Dr. Sanyal, once again. And now, may I request the convener of outreach program of Calcutta Statistical Association, Dr. Sanjeev Gupta, to give the vote of thanks.
Thank you, Subrata. So, as we draw the curtain of today's insightful and enriching lecture, it is both an honor and a pleasure to extend the vote of thanks on behalf of the CSA Outreach Program Committee. To begin with, I would like to express my deepest gratitude to our distinguished speaker and my dear friend, Dr. Nilotpal Sanyal. His engaging and intellectually stimulating talk has been a true highlight of the day. The clarity and depth of his insights have certainly left us with much to reflect upon. I am sure our students in particular will carry forward the learnings and perspectives gained today in their academic and personal journeys. My sincere thanks go out to the Honorable President, Secretary, Treasurer, board members, and the other esteemed officials of CSA. Your constant support, encouragement, and leadership are the driving force behind the success of this lecture series. A warm thank you to all CSA members for their enthusiastic participation and ongoing dedication. I also wish to convey my heartfelt appreciation to the respected professors, faculty members, dignitaries, and the student community, including graduate, undergraduate, and doctoral scholars, for their gracious presence and involvement throughout the session. A special mention must be made of Subrata Rana for his commendable anchoring. His skillful moderation helped maintain the energy and seamless flow of the event, ensuring meaningful engagement between our speaker and audience. I would also like to take a moment to recognize the incredible efforts of the CSA Outreach Program team. Your behind the scenes work, coordination, and attention to detail have played a crucial role in the smooth and successful conduct of today's program. Looking ahead with excitement, I am pleased to share that our next lecture in the series is scheduled for August. The exact date will be communicated very soon. We sincerely hope to see all of you there and look forward to your continued support and participation. With that, I bring this evening's program to a close. Thank you once again and good night. Thank you all.